I'm Bengt Norde. I am editor-chief of the quarterly reviews of Biophysics, and together with Giulia Palermo, also editor of the QRB Discovery. I am a semi-retired professor of physical chemistry at the Chalmers University in Göteborg, and I have also the role as chair of a research council in Singapore for one of the universities.
I'm Giulia Palermo, and I'm an associate professor in the Department of Bioengineering at the University of California, Riverside. I lead a computational biophysics lab that applies and develops computational methods to understand and engineer gene editing systems. We use physics-based approaches, including molecular dynamics, quantum mechanics, and other approaches to understand genome editing systems, and also artificial intelligence methods to engineer the gene editing systems of the future.
The journal is namely to report discoveries and observations and put forward material that may not be considered a complete story, but which points in interesting directions. I used to say that the paper in 1953 in Nature by Watson and Crick reporting on the structure of DNA is an example. You know, if you look at that paper, there is no new results. They are reporting on a helix that was deduced originally by crystallography by others, but they put forward the hypothesis that two strands are bound together by adenine binding to thymine with two hydrogen bonds and guanine to cytosine. So this was an intelligent guess based on the percentage of adenine being the same as thymine. But they had no clue about the selectivity because they missed the third hydrogen bond in guanine cytosine. So they've put forward this structure which first 30 years later proved to be correct by crystallography. But the paper and the speculation, if you may call it, had an enormous impact because it opened a mechanism for copying DNA, for instance, and so on and so forth. I'm not saying we want Watson Crick papers, but we are open to intelligent suggestions and pointing in certain directions that could be of general impact.
QRB Discovery is a place where you can publish your biophysical story, so the new mechanism of action that you discovered through a series of biophysical techniques such as single molecule experiments, cryo-EM. This is also evident from a series of special collections that are now open in the journal, such as on single molecule biophysics, on nucleic acid therapeutics, computational biophysics. So everything that together can work to decipher a biophysical mechanism is welcome in the journal. And the journal, of course, also welcomes bioengineering of biophysical systems, which could be achieved through artificial intelligence methods, diffusion models, and many other methods that can lead to the engineering of improved biological systems. So I think that in the current scenario where artificial intelligence is becoming more and more important, and we are also assisting at a revolution in the field of experimental techniques such as cryo-electron microscopy, so the interplay between these techniques and how it is used to decipher biophysical mechanisms, this is absolutely welcome in QRB Discovery.
There are no borders. I think that's exciting me. Things are developing very fast, and you have to have an open mind and you have to go into various directions. So if I should take an example from my own experience, I have been interested in hydrophobic effects, hydrophobic interactions. And as you know, the reason why lipids fold into lipid membranes or the peptide chains folds into proteins is to a great extent due to hydrophobic interactions, which means that we need water present. And that's why we need water for all forms of life. And in the case of the genetic material, DNA is stacked together and stabilized to great extent due to the nucleobases stacking on the top of each other like a coin pile due to hydrophobic interactions. And nature is taking advantage of that and is using it to how to catalyze the opening of DNA under certain circumstances. And we have been looking at that and detected how hydrophobic agents could lower the energy, the stacking energy. And we think this is very important in the nature of the DNA. So this is just one example.
I think the vision of the journal, and Bengt can further comment on it, is exactly how the interplay between multiple biophysical techniques can lead to new discoveries in the field of biology. So what is exciting, I think, about discoveries in biochemistry and biophysics is how can we find new mechanisms and new ideas that can characterize biological systems that were not characterized before, and how this mechanism can be part of the next ideas in science. So discovering mechanisms that can go in books. For instance, if we would open any book in biochemistry, we know now how about DNA, we know about RNA, we know about transcription, we know about translation. Of course, many things have to be discovered, but there are new mechanisms that we want to discover, and these new mechanisms will be part of the knowledge of the future. So I think this is the most exciting thing of our scientific journey, and is also, I think, the vision of QRB Discovery, the discovery of new mechanisms that will be in books. Students will read them, students will study them, and will build new knowledge.
I think an advantage of the way QRD is pursued is that it's a possibility for researchers who have got a bright idea and who are afraid of becoming scooped during a lengthy reviewing process. If they can show, if they can prove that this is excellent science, we will publish fast, and this is, I think, a strength of a journal.
The interplay between different techniques, different approaches, all together to discover new mechanisms and engineer new biological systems. So this is a little bit, I think, on my perspective, the science of the future. So in the future, it's unlikely that a single PI will be able to master all the biophysical techniques, because you specialize during your PhD in computational chemistry, in single molecule experiments. Every technique is so difficult to be learned, but the interplay between these methods, the interdisciplinarity, will make the science of the future and the scientist of the future a little bit like Leonardo. So Leonardo was able to understand different techniques and get the best from art, science, and was a great inventor because he has put together architecture and art and engineering. So I think in the same way we see discovery.
Well, we have started a number of projects where we look into new areas, and I think they provide good examples of where we want to go, and this is just the beginning.
A special collection on single molecule biophysics, another special collection on nucleic acid therapeutics, which I think currently is transforming medicine. If we think at what we can do with RNA, is absolutely amazing, is opening avenues that we couldn't think before with traditional medicines. So I think that there are so many areas, and we live in a time in which science is living a new renaissance. So So especially for nucleic acid therapeutics, I think is a field that is very important, and we welcome manuscripts in this field.
I think what Giulia just said indicates how important the editorial board is. We can actually be steering the science by identifying important problems and get people to suddenly realize that they are sitting on the answer. So I think this is something that makes QRD unique.
Because it's gonna be fast, it's gonna be very high visibility. What is very important, the journal has a great attention to the diversity in the corresponding author. Many papers have been published by women corresponding authors, and we think this is very important for pursuing science through a diversity of thoughts that actually benefits biophysics and science. Well, everybody can read the paper, high school students and young scientists that are getting interested in biophysics. They can learn from the papers directly. I think it's absolutely beneficial to publish open access to enable the scientists of the future to take their path, to read about our work, to get excited about it. I think this is the greatest benefit of open access.
My hope is that will be a journal that will be very general. Biophysics is far-reaching, but it will depend on the collaboration between so many fields of science. I think that QRB Discovery could be showing the way to go, how you could combine the various abilities of various technologies and scientific methods that leads to breakthroughs that are at par with the most important other journals. But the generality, I think, is very important, that we cover everything that could contribute to solving the mysteries of biology.